Hello everyone, myself Nalini, working as Assistant Professor at Maharani Science College for Women, Bangalore. Welcome to our new session. In this session, I shall be covering the enzyme kinetics. Enzyme kinetics is the study of rate of enzyme catalyzed chemical reactions. How exactly the rate of enzymatic reaction depends upon the concentration of the substrate. That is the main idea about this topic. In enzyme kinetics, the reaction rate is measured and effects of the varying conditions of the reactions are quite investigated. Here, the several places we see the rate of reaction, but here the enzymes are effective because they increase the rates of reaction. Enzymes are very much important in the biochemical reaction. The rate of reaction is regulated by the environmental conditions of the existing cell. Inside the cell, when the enzymatic reaction is taking place, catalysis is taking place, how exactly the need of the substrate, and if the substrate concentration keep on increasing, then how it is not going to affect the rate of the enzymatic reaction once the complete saturation of the substrate towards the active sites of the enzymes takes place. Once the saturation point is reached, then there is no effect on the increase in the concentration of substrates. That is the main thing that we are going to discuss today under the heading enzyme kinetics. We all know what is kinetics. Kinetics is the movement of the molecules with respect to substrate molecules, how exactly they are going to move and bind to the active site of the enzyme, and it establishes the enzyme substrate bond or enzyme substrate complex. That complex is the transitional complex or intermediary complex, which is very essential in the conversion of the substrates to products. Especially enzyme kinetics tells us about the importance of this intermediary state. Our enzyme substrate complex formation is quite important in all enzymatic reactions. The velocity at which the enzymes combine with the substrate molecules, and then they are going to convert into enzyme substrate complex. The velocity, the rate of the reaction till the formation of the ES or enzyme substrate complex, we take it as the K1. That is the velocity constant for the formation of the enzyme substrate. For example, in a NAD dependent dehydrogenases. Dehydrogenases are the enzymes. We all know NAD, that is nicotinamide adenine dinucleotide, will be the cofactor which is assisting this enzyme. Here we can observe one more thing. There are certain reactions where one substrate is going to give only one product, and it is not by rule, there is no such rule one substrate should give only one product, but we can also expect more than one products. Okay, we can take the example glycerol 3-phosphate dehydrogenase is an enzyme which is going to give two products out of three substrates. Like that, the number of substrate need not be equal with the number of products that are obtained after the enzymatic reactions. Likewise, two substrate, two product reactions are also there, as in case of NAD dependent dehydrogenases reaction, and one substrate, one product reaction. Very good examples are the enzymatic reactions associated with the enzyme isomerase, triose phosphate isomerase, triose phosphomatase. Like that, the isomerase based reactions are usually one substrate, one product reactions. So the one substrate need not always give only one product. There are variations in this. This is the standard graph that goes with this topic. Each and every slide text describes this particular graph. Here it is the x-axis, we are taking the substrate concentration. When the substrate concentration is 0, that we take it as V0. When the substrates are just initiating in the reaction and they are just approaching the active site of the enzyme, then the V will be 0. That V0, this is the point where we can see the substrate concentration is 0. And when the substrates are going to bind with the active site of the enzyme, the curve increases. That means the conversion of the substrates to products begins. When it completely reaches the formation of the products, we call it as Vmax. And intermediary point, we can see where exactly the curve intersects when it is going to frame a right angle with the x-axis and y-axis. This particular intersecting point, we call it as half the way to Vmax, half the way to convert into products. That is called as the formation of enzyme substrate complex, or intermediary state can be seen as of Vmax. This point will tell us about a constant called as Michaelis-Menten's constant, or Km. So the whole topic goes with this curve, how exactly the rate of enzymatic reaction with respect to substrate will be operating. Let's move on to the next one. See, when the enzyme binds multiple substrates such as dihydrofolate reductase, enzyme kinetics can also show sequence in which the substrates bind and sequence in which the products are released. For example, an example of enzymes that bind to single substrate and release multiple products. Very good examples are proteases, which cleave the protein substrate into two polypeptide products. In the same way, we can take the non-reducing sugar, that is sucrose. In presence of the enzyme invertase, it is going to give the products of reducing sugars like glucose and fructose. In other ways, two substrates together, in case of the enzyme DNA polymerase, linking a nucleotide to DNA. The two substrates giving out the product is individual product. That is also possible. Although these mechanisms are often a complex series of steps, there is typically one rate determining step. Even though the DNA polymerase involved reactions is many step enzymatic reaction, but there is one rate determining step that is very important. Here the rate determining step is considered as to explain the overall kinetics. That actually determines the overall kinetics of the reaction. This rate determining step may be a chemical reaction or a conformational change of the enzyme or the substrate, such as those involved in the release of products from the enzyme. Knowledge of the enzyme structure is very helpful in determining the kinetic data. How exactly the structure of the enzyme is, whether it is taking the help of any cofactors, all those things determines the kinetic data. For example, the structure can suggest how substrates and products bind during the catalysis and what changes occur during this reaction, and even the role of particular amino acid residues in the mechanism. How exactly the amino acid residues are going to act upon the intermediary state. See, some enzymes change shape significantly during the mechanism. In such cases, it is helpful to determine the enzyme structure with or without the bound substrate analogues. Enzymes, they themselves are going to change their conformation in order to cooperate the enzymatic reaction. Not all biological catalysts, they are proteinaceous. We all know there are RNA based catalysts such as ribozymes. These are very essential for the cellular functions such as RNA splicing and also the protein synthesis or translation reactions. The main difference between ribozymes and the enzymes is that the RNA catalysts are composed of nucleotides, but enzymes are composed of amino acids. There in ribozymes, we can expect the pile of nucleotides which are assisting in the enzymatic reaction. These ribozymes perform a more limited set of reactions, although their function mechanisms or the kinetics can be analyzed and classified by the same methods. The reaction catalyzed by an enzyme uses exactly the same reactants and produces exactly the same products as uncatalyzed reactions. But the mode of reaction, the quickness of the reaction or accelerated state of the reaction differs from uncatalyzed to catalyzed reactions. Like other catalysts, enzymes do alter the position of equilibrium between substrates and the products. However, unlike uncatalyzed chemical reaction, enzyme catalyzed reactions display this saturation kinetics. How exactly the active sites will be present in the enzyme, that cannot be expected in uncatalyzed reactions. For any a given enzyme, concentration and for relative low substrate concentration, the rate of reaction increases linearly. When the V0 is there, then if the concentration of the substrate is increasing, if it is going on increasing, then we can see the conversion of the enzyme still the conversion of the enzyme substrate complex, it will be increasing linearly. Once the enzyme substrate complex is done, then the curve goes plane and it is going to be steady state after the formation of the products. So the enzyme molecules are largely free to catalyze the reaction, and increasing substrate concentration means an increase in the rate of reaction at which the enzyme and substrate molecules encounter each other till the formation of Vmax, that is till the substrate intermediary state formation. There was a dotted line in that graph, no? That till that point it will be increasing. However, at relatively high substrate concentrations, the reaction is asymptomatically it approaches the theoretical maximum. The enzyme active sites are almost all occupied by the substrate. Once all the active sites are occupied, that results in the saturation point. In the enzyme, there is no space, there are no free active sites for the substrate to come and bind. And the reaction rate is also determined by this intrinsic turnover rate of the enzyme. How exactly the intrinsic, that is intermolecular efficiency, the amount of active sites or the number of active sites which gets saturated. So we can call it as intrinsic turnover rate of the enzyme. The substrate concentration midway between these two limiting cases is denoted as the constant, that is Lineweaver-Burk's constant, that is Km. This Km, thus the Km is the substrate concentration at which the reaction velocity is of the maximum velocity, that is the enzyme substrate complex formation or intermediary state. And also here in this context we need to discuss two most important kinetic properties of the enzyme. How easily enzyme becomes saturated with a particular substrate, that is one point, and the maximum rate it can achieve in a reaction, in an enzymatic reaction. Those are the two most kinetic properties of the enzyme. How quickly it gets saturated, how quickly it recognizes the substrates and how it binds with each other. And next one is the maximum rate it can achieve, how quickly it converts that enzyme substrate complex to the fairer products. Okay. The enzymes with single substrate mechanisms include isomerases, as we already saw. Single substrate, single product isomerases like triose phosphoisomerase, diphosphoglycerate mutase, and intramolecular liases such as adenylate cyclase, and also hammerate ribonuclease or ribozyme. So, or RNA liase, it's also called RNA liase. These are the examples for single substrate reactions. So, based on this number of substrates, we can classify enzyme substrate reactions into two types, that is single substrate reaction and double substrate reaction. Here in single substrate reaction, we can see substrates join with the enzymes at once make the AB enzyme complex, this complex, and then that is going to convert into the products C and D, and enzyme is released. At once it takes place. But in case of double substrate reaction, it is also popularly called as ping pong mechanism or ping pong reaction. Here what happens, the one substrate will first initially bind to the enzyme. We can take the example here. Here glutamic acid is the first substrate which is going to bind with the enzyme, that is pyridoxal phosphate, and also the coenzyme pyridoxal phosphate, which binds with glutamic acid transaminase. This glutamic acid transaminase is going to give the first product, that is alpha ketoglutaric acid, and it becomes that the enzyme becomes the carrier of the amino group. Since it is a transaminase enzyme, so it is going to remove the amino group from the first substrate, that is glutamic acid. Glutamic acid is going to leave or give the one amino group. That amino group will be carried by the enzyme, and in the next case it is going to give us the amino group to the next substrate, that is the second substrate in this double substrate reaction is oxaloacetic acid. Oxaloacetic acid is the second substrate to which the amino group will be transferred, and it is going to make the new amino acid, that is aspartic acid, and enzyme with the pyridoxal phosphate, it is liberated without undergoing any change. This is the double displacement reaction or ping pong reaction. So let us study what is Lichaelis-Menten's kinetics, and it is also popularly called as Lichaelis-Menten's equation. He discovered it in 1913. As enzyme catalyzed reactions are saturable, their rate of catalysis does not show the linear response to the increased substrate. After once it reaches the saturation point, if the substrates are increasing, substrate concentration is keep on increasing, then there is no change in the rate of reaction, since the active sites get saturated at this half Vmax point. So let us see how exactly the initial concentration of the substrate. If the initial rate of the reaction is measured over the range of substrate concentrations, the initial reaction rate increases. The substrate concentration initially, if you put the substrates from V0 till the off Vmax, then the rate of reaction will be increasing. But however, when it reaches the initial state or the intermediary state enzyme substrate complex is formed, then there is no change. That is the actual idea of this Michaelis-Menten's model of the single substrate reaction, and also it is true in the double substrate reaction. The concentration at Vmax, increase in the substrate concentration does not cause any change in the rate of the reaction. It will be steady state. Here the rate of reaction becomes very much dependent on the formation of the enzyme substrate complex. That is very important. And the reaction becomes a unimolecular reaction with an order of 0. From V0 till the off the way it will be catalyzed in the formation of the enzyme substrate complex. Though the enzymatic mechanism of the unimolecular reaction can be quite complex, there is typically one rate determining step. Even though it is quite complex process till the formation of the enzyme substrate complex, there will be one rate determining step. That step allows the reaction to be modeled as the single catalytic step with an apparent unimolecular rate that is called as kcat. That is the kinetics at which the catalysis takes place. If the reaction path proceeds over one to several intermediates, kcat will be the function of several elementary rate constants. So we can even see from enzyme and binding of the enzyme to the substrate giving out the intermediary product, there will be K1, and from intermediary enzyme substrate complex changing to products, that will be the K2. Likewise, there are so many constants we can take in an enzymatic reaction. The apparent unimolecular rate constant, that is kcat, is also called the turnover number and denotes the maximum number of enzymatic reactions catalyzed per second. So the formation of the enzyme substrate complex will be the final turnover constant that we can take, because that is going to decide. There is no further scope for the substrates to add to the enzyme. That is going to decide of the way so many products are going to form, or that so many products can be expected from this reaction. So we can call it as the turnover constant. We can see the reaction here. Michaelis-Menten's equation describes how the initial reaction rate, that is V0, depends on the position of the substrate binding equilibrium. How exactly? And the rate constant K2 will be the Michaelis-Menten's next step constant, that is the conversion of the enzyme substrates to the products, that will be the K2. So we can take this simple equation V0, sorry for this. So V0 is equal to Vmax substrate divided by Km plus substrates, where Km is equal to K2 plus K1 divided by K1. That is equal to dissociation constant. Kd will be the dissociation constant here. Km is equal to K2 plus K1 divided by K1. That is approximately equal to Kd, that is the dissociation constant. So Vmax is equal to kcat, that is the complete catalytic step, the important catalytic step in total with the enzyme concentration, that is E tot. This is the final Michaelis-Menten's equation according to this reaction which takes place during the catalysis. This Michaelis-Menten's equation is the basis for most of the single substrate enzyme kinetics. Okay, one more important note we need to discuss here. Two crucial assumptions which underlie this equation apart from the general assumption about the mechanism only involving no intermediate or product inhibition. Here we are concentrating only on the substrate, but there is no inhibition which is come and bound to the enzyme, and no allosterity, how exactly the feedback mechanism or the product itself will come and bind to the active sites, and there is no scope for the substrates to bind. We are not concentrating on these two aspects, even though that can also happen where the reaction is totally stopped and no products are formed. The first assumption is the so-called quasi-steady state. That is when the inhibitors are going to come and attach to the active sites, or pseudo-steady state hypothesis, when the inhibitors are attached and we cannot expect the products to be formed. That the concentration of substrate bound enzymes, and hence also called as unbound enzyme. The enzyme is not bound to the proper substrate, it has bound with the inhibitor, so it can be also be called as unbound enzyme. Changes much more slowly than those of the product and substrate. Thus the change over time of the complex can be set as 0. Here DES, enzyme substrate complex, over time, that is the dissociation or the time required for the conversion of the enzyme substrate product to enzyme substrate complex to product will be 0. The second assumption, when it is allosteric, is that the total enzyme concentration does not change over time. That is, E tot is equal to E plus E. There is no scope for the substrates to bind, but it has bound with the product, so it will be always constant. There is no formation of the products anymore. So Lineweaver-Menten's constant, or Km, is experimentally defined as the concentration at which the rate of the enzyme reaction is halfway done. That is till the formation of the ES, or intermediary enzyme substrate complex, which can be verified by substituting S is equal to Km. So substrate concentration is going to decide this Lineweaver-Menten's constant when it completely saturates the active sites of the enzyme. Lineweaver-Menten's equation, and can also be seen graphically. And very important, one more important thing, as we saw in that Lineweaver-Menten's equation, that is KD, dissociation constant. How quickly the EM, or enzyme substrate complex, is going to convert into product, dissociation constant. If the rate determining enzymatic step is slow compared to the substrate dissociation, then K2 is less than K1. That is, the K2 is the second step, conversion of the ES to products will be lesser than the velocity of the formation of the enzyme substrate complex, that is K1. K1 is the first step, formation of the enzyme substrate complex. That velocity is greater than the K2, that is dissociation of the enzyme substrate complex to product. That velocity is called K2. So the Lineweaver-Menten's constant is roughly the dissociation constant of the enzyme ES complex. Enzyme substrate complex should undergo dissociation. So mainly the Lineweaver-Menten's constant is the dissociation constant, that is Km. And also when compared to this plot, which is showing the curved graph or curved line, where exactly how exactly the V0 will reach as off Vmax and finally conversion into product, that is Vmax. But there are certain linear curves which are the plot versus the S substrates are there linear. Those curves which are non-linear to linear curves, there are certain hypothesis, that is Lineweaver-Burk plot, the Eden Hofstee diagram, and also Haynes-Wolff plot. These are the plots which are going to explain the same phenomena with a linear curve. The plot of V versus S, that is substrate concentration, above is not linear, it is curved as we saw. Although initially linear at low substrate concentration, it bends over the substrate concentration increases. That is what we saw in the last graph, right? Before the modern era of nonlinear curve fitting on computers, this nonlinearity could make difficult to estimate Michaelis-Menten's constant, that is Km and Vmax, accurately. So therefore, there are several researchers, they developed linearizations of the Michaelis-Menten's equation. So they, in order to understand better with respect to the computer graphics, they have made this curve linear curve. So it is first called as Lineweaver-Burk plot, and next one is Eden Hofstee diagram, and another one is Haynes-Wolff plot. So we can see those diagrams in our next slides. All these linear representations can be useful for visualizing the data, how exactly the intersection point is reached, where exactly the ES complex or intermediary complex is formed, and how it is going to change to products. But none should be used to determine kinetic parameters, as computer software is readily available that allows for more accurate determination of nonlinear regression methods. Even nonlinear plot can also be easily studied as the initial Michaelis-Menten's graph. So we can see AD Hofstee plot. Easily we can understand more generally Lineweaver-Burk plot, which shows the importance of measurements taken at low substrate concentration, and thus it can yield inaccurate estimates of Vmax and Km. So we will concentrate on Hayes-Hofstee plot, where V divided by S, Vmax, so we can see this Lineweaver-Burk plot first before going to the next one. So this will be the easy interpretation, how exactly the V0 point where it intersects this line, and how exactly the Michaelis-Menten's constant is reaching the Vmax. So Vmax, the formation of the Vmax, and off the way Vmax there is the intersection point that is going to decide the Michaelis-Menten's equation. Y is equal to mx plus c. We can see how exactly the Lineweaver-Burk plot is explaining, or it is also called as double reciprocal plot, is a common way of illustrating kinetic data. This is produced by taking reciprocal of both sides of the Michaelis-Menten's equation. How exactly the initial point reaches the halfway and finally to the Vmax. As shown in this linear graph of the Michaelis-Menten's equation, and produces the straight line with the equation y is equal to mx plus c with the y-intercept equivalent to 1 divided by Vmax, and an x-intercept of the graph representing 1 divided by Michaelis-Menten's constant, that is Km. So 1 divided by the velocity of this equation, Km divided by Vmax, that is going to give substrate plus halfway the formation of the enzyme substrate complex. So here also the same thing they are explaining, how exactly the intersect points of the initial and the of Vmax. At this point, the Michaelis-Menten's equation is relies upon the efficiency to get the products. Okay, naturally no experimental values can be taken at the negative of the substrate. Substrates are not going to take further by the enzyme, that is denoted by the negative substrates inversely proportional to the substrate. So lower limiting value will be always V0 at y-intercept point, where we can see this is the intersecting point where exactly the V0 can be achieved. Thus the x-intercept is an extrapolation of the experimental data taken at positive concentration. This particular intersect where we can see this particular thing where we can see there is increase in the substrate concentration for the reaction to takes place. That is how the Lineweaver concept is explaining the phenomena. Then we will come back to these two phenomena. So the same thing, Hidden-Hofstee plot explains how exactly the linear line can be used to see the Michaelis-Menten's equation. Hence, the whole plot is also similar to that of the Lineweaver complex. Lineweaver graph that we saw earlier. So what is the practical significance of this studying of enzyme kinetics or rate of enzymatic reaction? The study of the enzyme kinetics is important for two basic phenomena. Firstly, it helps us to explain how enzymes work, and secondly, it helps us to predict how enzymes behave in the living organisms. The kinetic constants, that is Km and Vmax, are critical to attempts to understand how enzymes work together to control the metabolism, making these predictions not trivial. Even the simple systems, for example, oxaloacetate is formed by malate dehydrogenase within the mitochondria. How exactly the simple enzymatic reactions will take place with utmost importance. Oxaloacetate can then be consumed by other enzymes, that is citrate synthase, phosphoenol pyruvate carboxykinase, and also aspartate aminotransferase. Feeding onto these in the citric acid cycle, we can see gluconeogenesis or aspartic acid biosynthesis takes place. So there will be interconnection between the two enzymes in the formation biochemical reactions, which gives further more products. That is how the substrate changes to the enzymes, and how exactly the substrate binds as and when the enzyme substrate complex frames, and that is going to convert into the different products. Being able to predict how much oxaloacetate goes into this pathway requires knowledge of the concentration of the oxaloacetate as well as the concentration and kinetics of each of these enzymes. How exactly the initial concentration of this formation of this oxaloacetate, the primary product, is going to rely upon the formation of the further products of the sequential biochemical reactions. That is how the Michaelis-Menten's equation helps us to know how exactly the enzymatic reactions takes place in the living organism, and also it explains how enzymes works upon by jumping from one substrate to another substrate. The enzymes are going to give the maximum products in the biochemical reaction. So this is the reaction velocity. It will be 0 at this point, initial point. It keep on increasing. Initially we can take it as the linear curve, but later it's going to give the curved line to reach the Vmax. Already we saw this Lineweaver concept, and now it is the time to summarize the topic. Please remember, it is very easy. Enzyme plus substrate is going to frame a intermediary complex, that is enzyme substrate complex. So the formation of this E plus S to ES will be the first step. The velocity of the forms, or the speed of formation of the ES, is taken as the constant, that is K1, and conversion of this ES to product EP, that is one more intermediary step, that is enzyme still attached to the product, that is taken as the K2. The formation of the products is taken as K2. So what is Lichaelis-Menten's equation? That is Km, or the Lichaelis-Menten's constant Km is equal to V1 plus V2 K1 plus K2 divided by K1. That will be the initial Lichaelis-Menten's equation. That gives us the idea of the complete conversion of the substrate to product, E plus P. Two most important kinetic properties of an enzyme are how easily the enzyme becomes saturated, that is the K1, and how it is going with a particular substrate, and how it is reach the maximum rate to give the products. So V0, when the enzymes are still approaching the active sites, V0 is equal to Vmax of the substrate divided by Km plus the concentration of the substrate. So Km is equal to K2 plus K1 divided by K1. So what is that is approximately equal to Kd. This is the dissociation concept. Dissociation of the enzyme substrate complex. Intermediary complex goes dissociation to give the products. So Vmax is equal to Kcat plus E-tot. That is total enzyme is going to get catalyzed. The very major step, catalytic step, will be assisting in the conversion of the ES to products. That is the simple Lichaelis-Menten's equation. It is all depend upon the enzyme formation of the enzyme substrate complex, which relies on the conversion of the substrates to products. So this is very, very important. Off the way to Vmax is called as off Vmax. That is going to decide no more substrates can be accommodated by the enzyme. This is the deciding factor. So many products are formed in the enzymatic reaction. That is well explained in all these plots that you can go through. These are some of the references for this topic. So hope all of you understood the Lichaelis-Menten's equation with the curved line, but at the same time there are other concepts like Lineweaver concept, Einstein's concept, etc., that can be studied for better understanding. Hope you all follow this particular session. Go through it slowly. It's quite lengthy and texty, and you can refer the textbooks to understand better. So it is very, very easy. The rate of enzymatic reaction is decided by the concentration of the substrate. When the V is 0, that is the velocity of the enzymatic reaction is 0, where the substrates are still has to bind to the enzyme. Initially it is going to rise in the graph because there is accommodation, still the substrates can accommodate into active sites of the enzyme, there is scope for the production of more products. But when the off Vmax is reached, that is the saturation of the active sites takes place, and that is the deciding factor for the rate of reaction. That is what Lineweaver-Menten has expressed in this equation. Vmax or Lineweaver-Menten's constant Km is equal to K2 plus K1 divided by K1, that is approximately equal to Kd, that is the dissociation of the enzyme substrate complex to products. Hope all of you understood. All the best. Happy learning.